And so now I want to start the project presentations to really highlight the work that you guys have worked on over the course of this weekend. So I went around talking to each of the project groups, but essentially the way that this will work is for every project, the entire team we asked to come up so that you all can be recognized. During the project presentation, the project leader, one of the project leaders, will be leading the presentation and giving the talk. But the project leader will be very explicit in giving credit to whoever worked on a particular part of their project so that everybody can be recognized. In order to get through this entire project presentation, because we have so many projects this year, we ask that you limit your project presentation to 7 minutes. Do these instructions make sense to everyone? Yeah. So I'll be timing every project presentation because we really want to try wrapping things up. At 1 minute left, I will raise my hand like this to show that you have 1 minute. And then for 10 seconds, I will show 10 fingers, just to let you guys know that it's time to wrap up. Does that sound okay to everyone? Okay. Does anyone have any questions? Okay, awesome. So let us start our final presentations. And so we will go in the order that you all contributed the slides. And so first, let's introduce the all-optical brain computing interface, led by Haley and Maxwell, with contributors of Ella and Paige. Can you all give them a round of applause for
Going on, everyone? Oh, sorry.
You're gonna take a picture first. Oh yeah.
All right, sounds good.
Please speak in front of the camera.
Sounds good.
Right.
Hello, everyone. So our project was aimed at an all-optical brain-computer interface designed at analyzing 2-photon calcium images to analyze the calcium fluorescent traces over the course of the imaging duration in order to elucidate the different neuronal signals and the circuits that are going on in response to some of the stimuli that were implemented during the experiment. And our first major task that we aimed to achieve in this was motion correction. The biggest issue that we faced in this stage of our project was that there was very little similarity from frame to frame. Unlike MRI images or other more static images which have clear anatomical structures which can be referenced between the frames in order to align them together, the 2-photon calcium images have very little features in them. There are no anatomical structures or very insignificant anatomical structures that can be compared between frames. And aside from that, the fluorescent signals, that is what we're aiming to quantify, change from frame to frame. So clearly we cannot use those two. So we worked to kind of come up with a different metric to kind of register the images, to kind of correct them, align all of our neuronal signals on top of each other so that we could identify ROIs and then track the progress of our calcium signals from frame to frame. So what we decided to do for this was we manually drew a couple seed points in each individual image or a subset of images that were representative of some constant characteristics from frame to frame, and then we used the positions of those features between them to define the transformation between the frames. And we kind of quantified the success of this protocol with two metrics. The first being displacement of each frame relative to our reference frame after motion correction. This was how far offset the two frames were from each other before and after imaging. We can see that in the blue is, or the red is before our correction. We can see that the images are a lot more offset from each other than in the corrected images. Our reference frame that we chose was the first image in the sequence, which was arbitrary, but the literature supported it. However, we did realize that this may not have been the most effective method based on our second evaluation metric, which was the correlation coefficient between each reference frame, or each frame relative to the reference frame. And again, you can kind of see the the red signals before our correction. We have relatively smaller correlation values that improve after the motion correction. But because of our choice of the reference frame being the first frame in the sequence, you can see that the correlation decreases as you progress from the first frame all the way through around to 700. So that was more a function of that, which is something that we will be looking to improve on in the future. And our second task was segmenting our image after our motion correction. This involves identifying relevant regions of interest that were including the neurons that we were interested in. And you can see the raw signal on the left. There's clearly a lot of noise there, clearly very little anatomical features, as I mentioned earlier, but we can still see the neuronal signals that we're interested in. However, identifying them was a little trickier. What we chose to do for this was we thresholded the original image with the edge detection, filled in each of the continuous edges, and then used those to kind of identify our neuronal signals. And from the results, you can see that we were very successful at doing so. And our next steps for moving forward, there are a few things left to do before we can implement this in real research settings, namely our analysis, including our spike inference, actually using our ROIs to track the neuronal, the calcium traces from frame to frame. Also, eventually, we would like to develop a GUI to make user interaction with the program a lot easier, so that it doesn't have to be super programmatic and can be translated from platform to platform. And then eventually, we would like to translate this to real-time capabilities, having it be capable of registering the images as they are detected, and then using that as input to a digital cursor. So these are some of the things that we're aiming of doing in the future, but very excited with what we got done this week. Feel very successful. We would like to extend a special thanks to our two undergraduate contributors, Ella and Paige. They were very helpful in kind of deciding the path that we would take, and how would we register the images, what parameters would be important in identifying the ROIs, and we definitely would not have been able to do it without them. So big thanks to everybody else here. I had a great weekend, and I'm sure everyone else in my group can say the same, and definitely would like to do this event again in the future.
Next slide's already up. So now we'll have trial and error. The project leaders for this project were Ainsley and Adam, and the contributors are Sefika, Mary, and Malbreet.
Hello, everyone. So I'm presenting our project on trial and error, led by me and Adam, who can be here today, as well as Sefika, Mary, and Kamalpreet, who joined us virtually. So an overview of what we're trying to do is look at the effects of trial history in physical experiments. So as an example, one experiment that we used was a data set that we had from our lab where people were asked to discriminate the direction of motion in either an auditory condition or visual condition. So for the visual ones, there's a random dot kinematogram, or just a bunch of dots on the screen that'll either be moving left or right. And for the auditory condition, it's a similar thing where there's a noise burst that either moves to the left or the right. And when we're thinking about the effects of trial history and experience on a participant's current decision, there's lots of ways to analyze this type of data, but this can be generalized to any two alternative forced choice task. So we started just by characterizing differences in accuracy and reaction time based on the previous trial or the previous two trials. And then we progressively got more and more complex with this. So Adam, me, and Sefika wrote up some of the pre-processing scripts that we use to generalize these functions across a couple different data sets. And then Kamalpreet was also helpful in creating this heat map over here, showing response times by stimulus type, as well as coherence level. And then we kept going and making even more complicated figures here. So everyone here is really helpful in adapting this data set to a drift diffusion model and running different types of statistical approaches to analyze the data. And for example, we looked at the effects of cue if we asked the participant to focus on the auditory stimulus or the visual stimulus, which might have a greater effect on a person's responses. And then finally, Mary was really helpful in developing a documentation so that each of the functions that we created have like a description of how to use it. And hopefully this can help make our resource more accessible for any neuroscientists who do psychophysical experiments. Anything else to add? Okay. That's it.
Next, we'll have foundation model adapters for spectrogram latent features extraction. The project leader for this was Chang Li, and the contributors were Yamin, Rinxian, Anna, Claire, Mason, and myself.
Hi, everyone. My name is Chang, and the whole group is me, Yamin, Renxuan, Anna, Claire, Mason, and Richard. Anna and Claire will not be here today because of personal issues. One of them is hit by a flu. Yeah, we have accomplished a lot during these days. Our project is about leveraging foundation models, pre-trained knowledge on EEG and fMRI spectrograms, and differentiate drowsiness and alertness states and patterns. What do we have accomplished? First, all participants heroically has configured a deep learning environments. I run the training pipeline, so it is very impressive. And we have integrated one of the visual foundation models, that is CLIP, into a pipeline, and we did some basic interpretation of EEG and fMRI spectrogram. So everyone, when they see a EEG spectrogram, they know where it's alert and where it's drowsy. And we did some UMAP visualization of the output features extracted by our foundation model and its adapters. And we have a better understanding of the how latent space features cluster to each other and how would that improve our classification performance. And we also did a little bit investigation of SAM model. We have, the model is currently in training. Yeah, what we have found out is that for a CLIP adapter model, we trained a classification head, use cross entropy loss. For the current ROI we are selecting, we have an average F1 score of over 90% on testing scans. For the UMAP visualization on the right side of the screen, you can see the yellow dots that represents that is the alert fMRI hidden features, and the purple ones are the drowsy hidden features. Our CLIP adapter can successfully extract features that has big discriminability in the latent space. So that is something very good for differentiating brain patterns related to alert and drowsiness. And the chart over here, it is the overall performance of a model. On the testing data sets consists of 3 resting eye closed scans. We have a very high accuracy, recall, and F1 score, as well as precisions. Yeah, for later on collaborations, what we have in mind is that we can visualize the latent space with different or multiple ROIs integrated, and that is the brilliant idea from Richard and Mason. And also, we can integrate the CLIP's pre-processing pipeline to data loading, so that streamlines the process, and it'll be and so the model training will be more efficient in its memory and its speed. And also, we have integrated the SAM model, and it's currently in training thanks to the effort of me, Rinxuan, and Yamin, and rest of the other folks around here. As a project leader, I would like to conclude that I'm very impressed with every member in my group, because all of the people configure the environment in such a short time. I run the whole pipeline, and they have a very advanced understanding of what latent space means and how to increase the performance. Yeah, I'm very impressed, and definitely there will be more collaborations for our group, and thank you, everyone.
Next, we'll have OpenDIVE. The project leaders for this were Adam and Alyssa, and the plenty of contributors, Dr. Rorden, Johan, Minyi, Adam, Lucas, and Dr. Schilling as well.
All right. Well, so yeah, we were OpenDIVE, Open Diffusion Image Visualization for Everyone. And so we were working on creating tooling for accessible ways of viewing diffusion imaging. And so we sort of had two sides to this. First was creating a colorblind friendly version of the traditional red, green, blue color mapping that we use to describe direction in these images. And so Dr. Worden has this really cool visualization he put together for this idea. We had a lot of thoughts on different ways we could view this imaging. And the idea that we've come up with is this accessible yellow, cyan, blue mapping. And he's got this really cool NI view way that you can visualize those and see. We can see here with the red-green color blindness, it's a lot more accessible and available to those people for it looks very similar to us.
And I do tractography research, and I find myself spending a lot of time with trial and error putting together tractography visualizations. So we were looking to make a command line tool that we could easily visualize things like tractography and changes over time with different color maps and color bars. So we created this Python package that you can download, including data and things like that you can find on our GitHub. And we can see all of these different regions of the corpus callosum and all of these different arbitrary mappings that we might have based on different p-values or change over time or other effect size related metrics that you might want to visualize.
Yeah, and so this was really great. We really appreciate everyone contributing. I think we had a lot of people go from like 0 to contributor on diffusion imaging. And so yeah, Dr. Worden helped us with the visualizations for the colorblind mapping. Joanne got us on 40 images, multiple tractograms. Mia helped us with plotting the tractography. Adam helped us on documentation and started on diffusion tensors. Lucas helped us with examples and testing. Kurt Schilling helped us on tooling and inspiration for the project. Alyssa helped us implement these color bars, and I did some of the testing and merging. So yeah, thanks everybody.
Hey, thank you. Now we have dynamic neural decoding for adaptive neuroprosthetic control led by Daniel Woods with contributors of Julia and Mohammed. Julia and Mohammed are virtual today, so it'll just be me up here if you want to take the photo. Yeah, of course, of course. Yeah, so this project definitely wouldn't have happened without the help of both of them. I kind of color coordinated what each of us were able to take care of. So the overall goal coming in was we had these example recordings from our flexible electrodes in the lab in mouse visual cortex, but really that was it. It was just a long list of 32 channels of voltage differentials, and we're starting from scratch. So what Julia really worked on, if the cursor appears, you guys see this? Yeah, so she worked on spike sorting and curating. So you really have to do a lot of sanity checks when you have electrophysiological data to make sure that when you're filtering and trying to look for spike waveforms, it's not just noise. So she went through and used a template matching program called KiloSort. It's kind of an open source spike sorting software. And she identified some spikes of interest and then fed those through a visualization toolbox called Phi GUI. And with that, she was able to manually curate the waveforms of interest. And to kind of simplify our process, we really went from here to just looking at 2 trains of spikes. We took our most active channels from this recording. I should say like a big goal of this project was to allow for some form of a live neural decoding while the mouse might be performing this task. But we have recordings just from an anesthetized mouse in visual cortex. So we kind of did some work around to make like a post hoc decoder. So once these spikes were filtered and manually curated from Phi, we read them into just like a standard this is a Plexon system. They make different probes for the brain. They have different file configurations to help you read in your your data and get it into a more manageable format. So that was something that I just took our data, went back to the lab last night, and gave Muhammad back our series of the 2 most active spike trains from 2 different contacts along our flexible probe from the mouse recording.
and he performed a series of dimensionality reduction methods. So this is also all through an open toolbox called Data High that just takes a high dimensional electrophysiological data set, and you can select obviously like your type of dimensionality reduction method, your number of dimensions. He ended up going with a Gaussian process factor analysis. He also tried some PCA, and we're definitely going to mess around with like different forms, because when you have neural data, some things work as well, some things don't. Notice that for for GPFA that the variability between two groups of neurons could be better accounted for as just like a general PCA. So this visualization is just the first 3 dimensions of an overall 40 dimensionality reduction process. So that described for our 90% of variance between these two trajectories. And they just plotted them in 3D space. It's just kind of fun to look at how do these two sets of cells respond. This is over a 40-minute recording in anesthetized mice. So then the very last thing that I did is, I think it's fun to look at, but it's not necessarily exactly what I want it to be long-term. This is just a visualization that takes 2 sample trains of spikes, one controlling a cursor movement up and down, and then you simply just have a button that toggles like the polarity of the 2 directions. And what I did, because since the 2 neurons were our most active that I picked out, they were the easiest to perform our analysis on, they had pretty similar spiking behavior throughout the task. So really the cursor stays pretty still. So I just introduced like a little bit of random variance for the neuron that would be driving mouse direction towards the cursor. I would imagine this is like long-term when we're introducing this into a live decoder, that with an animal behind the wheel of the neural activity, other papers have shown this, that you can influence this direction. That's how neuroprosthetics work. So yeah, I'm really happy with everything our group is able to do. Moving forward, definitely introduce this to a live task that requires a lot different set of skills, but it was really fun doing this with our recordings we already had. And some different dimensionality reduction methods. This would mainly be to account for latency, because when you're doing this with a behaving animal, you want to make sure that the cursor drives in time. And yeah, that's basically everything that we did for the weekend. So it was a lot of fun, and our group was really great. So thank you guys. Thank you, Daniel. Next, we'll have validating DeepMReye. The leaders for this were Elton, Jin, and Zoha, and Dr. Vincent Bucher. And the contributors were Kim, Nerdy Lab, Nerdy Lab, and Tess, I guess, and Vanderbilt.
Hi, I'm Ashley from Land Lab, working with my fellow project leader, Ali and Zoha, and our great collaborator, Kims and Taste. She's not here today, and our PI Winston Boher. In our lab, we're using indoor eye tracking method while acquiring fMRI scan. Our goal in this BrainHack is there is a open source deep learning library called DeepMReye that is able to extract the eyeball voxel signal from fMRI scan and predict eye position. We had adults and children participants, and our goal is first to validate the accuracy of eye gaze prediction produced by DeepMReye for the adult participant, and then we want to see if the model can be used for children participants. This is because DeepMReye was training with adult participants only. And we encountered many problems over the last 2 days. Our first issue was figuring out how to extract eyeball voxel. We could not find the mask to extract eyeball voxel on their GitHub initially, so we began create eyeball voxel making masks on FR photo as simply by using mask to and manually coloring in where the eyeball voxel are. While working on this, we're able to find DeepMReye mask, and we're successfully extract the adult eyeball voxel. And in this, heat map is their eyeball voxel, how their eyeball voxel looks like. With the red color indicating the high intensity and gray color indicate lower intensity of their eyeball activation. And in this histogram, it represents the distribution of voxel intensity, volume, or activation with the eye masks. And the line plot could show and this line plot show a time series of both signal extract from eye masks to visualize the temporal dynamics of extract eyeball voxel signal. And this plot here predict gaze position of x and y coordinates over functional volumes. DeepMReye generates the predict gaze coordinate from our fMRI data. We also run children's. We also try to extract children's eyeball voxel volume, but DeepMReye didn't work very well on children's. And then we moved to changing dimension of our eye gaze data so that it could match our expected dimension that we could have to fit into the model. We also, at this point, download the training weighted from DeepMReye paper. And now we are working on to fitting the correctly formatted data into the actual model to predict the eye gaze. And then our last time we're able to comparing the prediction with actual eye gaze data. Thank you all for listening, and especially for our teammate Kim, Ali, and Zoha, and our PI Lindsay Border. And also thank you so much for the BrainHacks organizer. We have a great time. Thank you.
Next, we'll have brain state modeling. So the leader for this is Bruno, and contributors were Pam, David, Manish, and Lily.
All right. Hi, everyone. David is unfortunately sick, but still hacking from home. Yeah, we had a really great team, and I think something really cool was that we went in a couple different directions because people had some different interests in the group. So just as a reminder, the project was mainly based around this model that takes intracranial EEG data from epilepsy patients and tries to model that data in the form of brain states, which are like high dimensional embeddings. And we kind of look at those as point clouds by projecting down to a lower dimensional manifold. And really what I came to this event with was the sleep stage data and trying to characterize the ability to capture different sleep stages with these embeddings. Pam and Lily took the lead on this by characterizing a different dimensionality reduction technique called T-SNE, which is kind of more geared towards local structure, whereas what our lab had been using, which is PACMAP, is more about preserving global structure. And it was really interesting. I think what we learned was that you can still kind of segregate these different sleep stages with T-SNE, which is useful for our lab to know because PACMAP is a little bit more computationally expensive. So it's good to know that T-SNE also captures these differences. And also, Pam did some 3D representations of that data. And as you can see, it's pretty interesting because, for example, the blue dots here, which represent the wake stages, although it might seem as if there's two different clusters in 2 dimensions, like here and here. When you look at it in 3 dimensions, you can see that they actually do kind of align a little bit more consistently, and that when you project down to 2 dimensions, you start losing more and more of that representational structure. As you can see here, that even though, I mean, the relative positioning of these is always arbitrary with dimensionality reduction techniques, but there's still a lot of utility in representing it in as high dimensions as we can, conceive as humans. And then the other direction that we went in, which I actually wasn't really planning on, pitching for this project, was actually applying this data for downstream application in a closed loop neuromodulation system. And so ultimately, the way that would work is we would be monitoring a patient's brain state in real time and kind of mapping it onto this high dimension or this point cloud. And the idea is that we would be able to anticipate when that brain state is kind of trending towards a seizure. And what that would essentially entail is being able to take a series of brain states and then make a prediction of what the next brain state will be based on that previous series of brain states. And so Manish, David, and I worked more so on building a transformer that took in segments of brain states and then made a prediction on the next brain state. David kind of took the lead on building that transformer. Manish made these really nice plots. And as you can see, the red trajectory is the trajectory of brain states that the patient actually took during a period of time. And then the transformer was able to predict a relatively similar path, which we denote in that yellow. As you can see, this holds up even across brain state trajectories that span across the entire latent space, which is pretty interesting. These first two plots that I'm showing were based on one-shot predictions, which is basically purely based on the ground truth. And that would essentially require us to always be making a prediction based on data that we already know. What would be more interesting is if we can make predictions that are operating on what the model has already predicted, which allows us to kind of predict a little bit further ahead in time. And as you can see, a little bit more noisy, but the model was able to hold up. This is a case where the model kind of started over here, came down, and actually went back. And the model predicted, kind of straight away a little bit here, but ultimately was able to track the trajectory back to where it started. And this is just another example. So, I mean, this was really cool. I mean, this is not something that I was expecting to get out of this event, but this was 100% something that our lab had been meaning to get towards, because ultimately there's, I think, real utility in this data. And so we started kind of getting towards that. But yeah, had a lot of fun and really enjoyed the event, and shout out our amazing team.
Thank you so much. Next, we have less confusion in diffusion. The project leaders for this were Nancy, along with, I think you'll introduce them as well. Yeah, but we got a lot of contributors on this one.
Okay, perfect. Oh. Sweet. So myself and Elias were the project leaders for this, but we also had Shiyu, and Andre and Shansita, and then reading with us virtually are Trent and Eric and Gaurav. Yes. Okay, so we started this with the goal to create a image to text bot to help point out the imaging artifacts and acquisition issues that need to be fixed in a given image slice. So the issues that we focused on were slice dropout, the top of the head being cut off, and having poor resolution. There's obviously many more issues than this for imaging, but we really just wanted to try and focus on groups that we thought were easily separable. So an overview of what we were able to accomplish this weekend. First, we curated a data set of over 3,000 image slices and labels. Here I have like myself and Gaurav in parentheses. We all contributed to each of these steps very heavily. It was kind of crazy, but I think we got a lot of really good overlapping experience. But I'm going to point out who really shined for these tasks. So after that, we searched the literature and Hugging Face to see what pre-trained models might fit our task. So what are we starting with? What have people already put together? And here Elias really shined, having experience with language models before this. After that, we basically did some model selection, which we'll talk about in a second, and got these pre-trained models up and running on our devices. And here, Shiyu and Shansita really got the ball rolling for that. After that, we started just doing the zero-shot inference on our data set. So we took the model, it already had some example data, but we wanted our own data loader and to integrate the data set that we were actually looking to work with. And after that, we decided we needed to fine tune with our data from there. There, Trent and Eric really shined. And then after that, we had some mixed results, but we did put everything together into a GUI. So there is some like user input output that we have to show you guys. So right now I will pass it off to Shiyu to talk a little bit about our model selection.
So we picked our models based off of whether it's well documented, whether it's easy to apply, and if it's fine-tuned for medical images, which is what our application is. So we tried out these 3 models, and we mostly focused on the first one, Biomet Clip. It is a large vision language model fine-tuned on biomedical images using 15 million figure caption pairs from biomedical image research papers. So here I think it's showing the some zero-shot outputs from Biomet Clip. So these models takes in the prompt. This image has the following issues. So basically, we're trying to identify what kind of artifact or issues that are there with these images, and the labels for some of the examples that we're going to show are whether it's field of view issue, cutoff at top of brain, slice dropout, or low resolution. Here are some examples of these issues. So this is also some zero shots results from one of the more advanced vision language models fine-tuned on biomedical medical imaging. So this model takes in the image. In this case, it's the corrupted DeepMReye brain image with slice dropout issue. And it also takes in the text prompt. And here we try to describe this image. And then it gives a text output in response to this the text prompt based off of the image that you give it. So here you can see that like for this image in the middle, it just failed to recognize that this is a brain. And then for the first one and the third one, it was saying that this is a brain, but either giving it a wrong label or wrong diagnosis or the wrong imaging modality. So improvement is definitely needed.
Yeah, so looking at those, we kind of decided that we needed to like impose our own workflow on these models because it wasn't, you know, exactly what we were looking for. And some of them were fine-tuned more for like diagnosing a patient. And so it was telling us about like multiple sclerosis, and these patients were all healthy, so not have multiple sclerosis. So we started, we took the Biomed clip and we started to fine-tune it. So we're going to go through this training curve and kind of show you like 3 snapshots of what the outputs look like if we use that model. So this first one, I will point out that the loss we used for fine-tuning was the cosine similarity. This is a big thing that I learned personally. This loss is trying to optimize the embeddings between the image and the text label. Okay, so first off, if no training is done, but we impose our own workflow on it, this is what the GUI looks like. You provide an image, you load the image, and then you ask the question. And we output the following potential issues were identified, and it's going to tell you a list of the issues and the confidence score, or essentially like the probability that it belongs to that class. So at first, it's saying that both of these are like 100% missing slices. And while that's true, for the second one, we quickly realized that it's just saying that for all of the examples. As we continue to train, the model starts to understand that there's multiple different classes. So in this one, at around 5 epochs, it's saying, oh, missing slices, but also it could be motion artifacts. Finally, at 20 epochs, it's understanding that we have like 4 to 5 different classes that it could be. Now, it's still pretty much saying that everything is like falling under the like missing slices or like motion artifacts. So we did not get to the level of like effectiveness that a full product would do. But we're still very proud that we were able to get this workflow going and that we were able to identify these different classes. So overall, the takeaways were that the documentation is really important. So I think we did a good job on our own GitHub on like making sure that we were documenting everything. There were really cool models out there, but sometimes if it wasn't well documented, we could not use it. Also, language models are data hungry, so I think just adding in a bunch more data would be would really improve our results. Overall, I'm super impressed with everybody on the team and what we were able to accomplish. So thank you to our organizers and to the team.
Next up, we have multi-site fMRI harmonization. So this project was led by Isaac with collaborators of Daniel, Kimmy, Oscar, Ella, and Dana.
If you edit them last minute, they might not be here, so we need to probably refresh that page. Is this the one?
Yes.
Okay. Oops. What was that? Okay.
Awesome, thank you. So, hi everyone, my name is Isaac and I'm the project lead for the multi-site fMRI harmonization project. And today I am joined with my awesome collaborators here in Daniel and Kimmy. And they've been working tirelessly throughout the both 2 days of the BrainHack. And we also like to acknowledge help from Oscar, Ella, and Dana, who have been super helpful with our project during the first day of the BrainHack. And so to give everyone a quick refresher of our goal for the project, basically we are trying to leverage 3 data sets that our lab has collected over the years, and we want to try to harmonize, combine the data in a meaningful way. And so the goal that we had was we wanted to use this software called Combat to remove between scanner variability for the different data sets. And so basically over the weekend, what the approach that we took was that we sort of all worked on similar goals together. So we were progressing through that project together so that everyone knows what's going on throughout each step of the process. And so we went through a tutorial of Combat to get everyone comfortable with using Combat, what goes in, what goes out of Combat, and the sort of structure that the program sort of expects. And after that, we worked on a pipeline to prep all the Nifty files and get them in the right format that we wanted to use for Combat, and spent a lot of time like figuring out like what the best way and like what specific features we want to use to harmonize. And ultimately we decided to look at harmonizing values at the ROI level. So our task is a visual rhyme task where the kids are asked to decide if 2 words rhyme or not in the scanner. And we chose 3 RIs of interest, specifically the inferior frontal gyrus, the left superior temporal gyrus, and the left fusiform gyrus. So we looked at these three ROIs. We took an average of the values inside and harmonized those values across different data sets. And after getting the harmonized values, we then saved them in a way that we can use them and applied a linear model to evaluate potential side effects. And so thank you. So this is, we did a linear regression model of the pre-Combat harmonized values as a function of age, sex, and site. And as you can see here, this is, and I forgot to mention, this is for the left inferior frontal gyrus. And as you can see in the statistical summary on the left, we do see a side effect for one of our data sets compared to the reference data set. But after we ran Combat, you can see that the side effect went away. And so this was an indicator of that Combat was doing its job. It was removing the non-biological variability in our data and keeping the important biological stuff that we're mostly interested in later downstream analyses. After that, we decided to plot the values to see like how the distribution of the data changed. And you can sort of see here that on the picture on the left, The third data set had the one in the blue had more variability in terms of the values for the mean ROI. But then after combat, a lot of them sort of converged towards with each other. One other thing I wanted to mention about this is the two data sets, the first two, the green and the red, these two actually use the same scanner, but they use different imaging acquisition parameters. So it was also nice to see that even though they use the same scanner, the values sort of were in the same range. But for the blue one, this one was actually a different scanner and a magnet strength of 1.5 T and not 3.3 T. And so seeing that combat works for scanners that are different and in terms of the scanner manufacturer and the strength was reassuring for us. And we ran this for 56 different subjects, and I think that was really awesome. Okay. In terms of our next steps, one trouble that we had during the BrainHack was that we had trouble running combat when we were interested in looking at the whole all the voxels within the RI mask, and that was because there were issues with missing values, and that was interfering with how the function runs. So Dr. Roden actually gave us advice as to how to deal with that. So I think as our next step, we will try to implement his advice and see if combat works. And we also wanted to evaluate how motion impacts the effectiveness of harmonization as well. So we want to see if motion makes a difference or not. And one other thing we want to do in our next step, because we are also collecting, currently we're also collecting data in our lab that we want to harmonize with the data set that we are working with over the weekend. And that data set has multi-band data compared to the non-multi-band data we were using this weekend. And so we want to also evaluate the effectiveness of combat on multi-band versus non-multi-band data. And lastly, we want to develop a fully automated pipeline for data cleaning, processing, and harmonization, get everything streamlined and ready to go. So this was my first BrainHack. It was a really nice experience, and I appreciate all the hard work my two collaborators, Daniel and Kimmy, have done over the weekend. And this was a great experience. Thank you.
So that wraps up our project presentations. Please, again, can everyone give another round of applause to all of the project leaders and all the project groups for their incredible contributions throughout this weekend. It is truly so impressive what you guys were able to accomplish in just a weekend. I'm speechless. Like this is absolutely awesome. And it's really shows, I guess, the power of collaboration and the power of accessibility and including everyone in diverse perspectives, which is really the message that we want you all to take away